All right, our task for the next hour then is to have a look at the assignment itself, to go through each of the parts of it, and then when we get to a page where there's some planning that needs to be done, you are going to write things in there. And the bottom of that page, it says about the procedure will be planned in class. Well, it's already planned, and I will show you the plan sheet. I might put that on Moodle if anybody wants to print it off and have a look at it, but it will be available in here during the practical sessions. And so I will go through, you know, when you walk into this room next week, into your practical session, what's going to happen. And we will then have a look at what needs to be handed in, because some of that you can be making a start on. You can do the introduction, and you can, you know, start having a look at, well, doing some preliminary practical work for yourselves. And then we will come back to that. The week after the practicals, I will have a session which is entirely focused on going over how to use Excel, and referencing, because a lot of the referencing I'm coming across in your first assignments, you're just not following the instructions. You're making up your own formats, not listing them in alphabetical order, stuff like that. So I've got to go over that again, because it's not, you know, it's contributing to grades in the first one a little bit, but it will become more and more important. So referencing is a critical thing. So first job then is having a look at the assignment. We've got issue date was last week, wasn't it? And you've got until the 6th of December. If you haven't got it completed by then, hand it in incomplete. If you've got a reason for it needing to take longer, put in an extension request. The forms are hanging in a thing on the plastic pocket on the notice board over there. The assessment criteria we've been through. We've looked at what you're being assessed on. 2.1 is about the introduction mostly. 2.2 is about the practical and the table and graph. 2.3 is about the mean and standard deviation. So the words you're looking for in that section are accuracy, to do with the mean, and precision, to do with the standard deviation. 2.4, misleadingly, only 6 words there, is the biggest part, analysing the data. And then analysing the actual success of the whole thing and looking for any sources of error or bias that are not things like, oh, well, another group might have done it differently. No, they didn't. We'll go through that. You've got 3 grade descriptors, 1 for understanding, so you need to be explaining the science, all right? This has got to have substantial science in it, level 3 science. Grade descriptor 3 is application of skills, which is partly your practical work and working in a team and possibly supporting people who've not been here for the instructions. It's partly about using IT, so it'll be about all the clever tricks with Excel, which you're going to have a go at tomorrow afternoon, which I showed you yesterday. And it's also about your skill in just generally writing up a good practical report and your analysis skills, that sort of thing. And then number 7, as usual, is the quality one, which is about the logic. You know, have you got it in the right sort of order, sequencing? Does it flow properly from one bit to the next? The general quality of your writing. Is it a good, very good, excellent response to the task? You know, my general impression. And so those 3 grades, of course, can be an absolute game changer. Regardless of what you've got on the first assignment, you know, you could completely change that with what you've got on this one. Of course, it could go either way. You know, you could get 2 distinctions on the first assignment, 3 passes on this one, and your overall grade's going to be a pass. So what you want, of course, is to go in the other way, but it's not easy, all right? And distinctions are a rare thing. Be warned. The descriptions of the grades are there, which won't mean a lot to you, but we just have to put them in. Now, the focus for this then is osmosis as a process, but it is, the actual practical is about Finding the water potential of potato tissue. That's even a reasonable title for the graph, actually. Finding the water potential of potato tissue. You can put a draft in, as ever. I can only give you feedback on whether you're passing okay and look at things like structure, style, spelling, grammar, referencing. Can't tell you anything about grading. All right, that it's just not allowed. Overall, it's about assessing your understanding of transport across cell membranes. So make sure you've done a bit of reading on that before you start writing. Your skills in carrying out practical investigation and your ability to process and present the results in an appropriate report. So you've got 1,500 words to do this. Add 10%, goes up to 1,650. Take 10% off and you're not doing enough, basically. Right, the background to the assignment is just reminding you about osmosis and the definition of it. Now, you can literally copy the definition into your introduction at some point. But the idea is, of course, that we're going to use this principle to try and find the isotonic point, the water potential of potato tissue. You're going to put some potato into hypertonic solutions with a lot of sugar in, hypotonic solutions with very little sugar in, and then somewhere in the middle there'll be an isotonic solution, which you won't necessarily use, but the isotonic means it's the same concentration in and out, really, same osmotic potential or water potential, so that water moves in and out at the same rate and you don't get any change in mass. So that's the principle behind it, and you can use information from this assignment brief to help you write the assignment up. The task then is to carry out the practical investigation using potato tissue and sucrose solutions of varying concentrations and therefore varying water potentials to estimate the water potential of potato cells. It's worth looking at different techniques beforehand, but you are going to be given a method, but if you look at other techniques, then you can evaluate the method that we use and compare it and criticize it. And, that can be a useful thing to do. So make sure you know what you're doing before you start. So practical sessions are next week, and we break you up into 2 groups so that you've got space to work, really, and so that we don't have problems with equipment. We do have to turn the equipment round quite quickly sometimes. So, for example, the group in here next Wednesday afternoon, there will have been the other group on Wednesday morning, so we have to turn the equipment round between the morning and afternoon sessions. They've got a big group and a small group, so we've put the small group on the Wednesday morning, so we've got less stuff to try and get ready for you. All right, it will take 2 hours. So be prepared. Now, this sheet you need to fill in, and this should then be material that you can use in your assignment. So first of all, we need to identify the variables so that we know what we're working with. So what will the independent variable be? What are you going to change, or what have we decided to change the values of? The sugar solution, the concentrations, but that's not actually the independent variable. The independent variable quite specifically is nothing to do with the potato. You are right, it's to do with the sugar solution. What aspect of it? The water potential. Yes, it's not the concentration, it's the water potential. Okay, so water potential of the sucrose solution. Now, you will be given a bottle of sugar solution, sucrose solution, which says on it 1m. The m is short for moles per decimetre cubed. And I know you don't know what moles per decimetre cubed are yet. We're not quite there. But it's a way of measuring concentration, and you're going to make different concentrations from that. You're going to make concentrations which are 0.1, 0.2, 0.3, 0.4, 0.5, and then you'll leave it at 1, and you'll also have water. But that's not your independent variable. It's a way of getting an independent variable, but your independent variable measures will be, the values that will be used are, and I'll write them up here for you. You're going to have 0, which will be what? Pure water. And then you'll have, in terms of sugar concentrations giving us the different water potentials, we have minus 260, minus 540. These are getting more concentrated. Minus 860. Minus 1120. I'm going to run out of space here. Minus 1450. Minus 3510. And my unit is? No. Yes, I was hearing it. Kilo. Kilo pascals. That's it. Well done. So kilopascals are the units for water potential. All right, now you will get a set of instructions for making these solutions up. So one person in the group is going to make up those sugar solutions, but you'll get the instructions on how to. So somebody with a steady hand and a good eye for measuring volumes. So what's the dependent variable then? Size of the potato? Or, what are you actually measuring? Mass of the potato, but what aspect of the mass? What aspect of the mass? Not surface area, no. What aspect of the mass? That is the same thing. All right, the aspect of mass is the change, but what aspect of change? It's the percentage change, yes. All right, hang on a minute. I thought they were going to be checking badges. All right, for those listening to the recording, we've just been interrupted. Right, our dependent variable then we decided was percentage change in mass of potato, or percentage change in potato mass. And how are you going to measure that? All right, in grams using electronic balance. Okay, now there are things that you'll need to keep constant in this. What sort of things will need to be constant, do you think? Temperature as far as possible. We know what it's like in here. Chances of the temperature being constant, not good. Yeah, it'll start hot, it'll cool down, it'll warm up again, who knows. But we would do our best. And if we can't keep it constant, we'll comment on that. But because particles move about at different rates at different temperatures, it would be important. So, temperature. Now, there might be some other things that we'll need to add to this as we go. What about the bits of potato? Same size as far as possible, so that each group is cutting pieces of potato the same size. Anything else that you can think of? It doesn't actually matter. You will make sure that you're all using the same concentrations, and you have to assume that everybody's being accurate making those up. Time, yeah. So you'd all need to leave them exactly the same amount of time. There is something else that you won't be aware of yet, but we're going to put it in here. And that is, when you've cut your potato pieces, you actually need to dry them before you put them in the solution. And then when they come out, you'll need to dry them again. All right, now you dry them before because you're going to dry them afterwards, because they'll come out really wet, won't they? So, how are you going to dry them? No. Paper towels. So what would you do? Put them in the towel and then pat them? No. Yes, you wouldn't pat them because that would put pressure on them. So what might you do? Roll them. Yeah, not a bad idea. So what you can do is to put the pieces of potato onto, and because they'll be like little cylinders, little sticks, you can put them onto a paper towel on the bench and just push the towel back and forth, and they'll roll about, won't they? And that will take enough moisture off that if you do it again afterwards, you'll be consistent. So it would be the drying technique. Yeah. We can't control that. We've got no control over that at all. Well, origin, yeah, the bag of potatoes, yeah, you could have that. So drying technique we'll put in. Yeah. Or, yeah, but if you just go for you know, if you go back and forth 4 times, if everybody does that, you're all, yes, that's the important thing. Origin of potatoes. What you tend to find these days is that, I don't know if you ever look at the labels on fruit and vegetable packaging, but they will actually name the farm sometimes on the label. Not on the, you know, the pack bit, but when you've got the individual label. So they will very likely come from the same farm. All right, so Jill will just buy a bag of potatoes. I think that's not absolutely certain though, because what we need is 11 big potatoes. Because what you don't want to do is to use more than one in a group, do you? So that's something I don't know if we can, yeah. So yeah, and trying to get them all out of one potato. So they need to be fairly big, and of course, well, I suppose if she gets baking potatoes, that's possible. But yeah, so there are things that we can keep constant. There are things that are much harder to keep constant. Yeah. Green potatoes. We wouldn't use green ones. They it's the ones that have come. Oh, right, okay. No, they have oxalic acid in the green bit. But I think the green is only chlorophyll. Because, sorry? Yeah, there's a small amount, but it wouldn't really hurt you. But if you ate a lot of green potatoes, it is toxic. But the green is, I believe, just chlorophyll, because potatoes are not roots, they're stems. Because, you know, if you leave a potato, it shoots, doesn't it? It produces leaves and things. A potato is actually an underground stem. So if it comes above ground, you leave them in the light, they go green, so I'm assuming that's chlorophyll. But they also produce what I think is oxalic acid. Oxalic acid you also find in the leaves of potato plants, they're toxic, and in the fruit of potato plants, which you never see. But potatoes will flower and produce little fruits above ground, but they're toxic as well. So, yeah, we just, there'll be supermarket bog standard large potatoes. So? Fruits on a potato. You don't, people dig up the potatoes before they form. But, you know, you've got the green plant, yeah? It then produces white flowers, yeah? Well, what do flowers turn into? Fruit. That's what flowers are for. Flowers turn into fruit. So potato plants will produce fruit. But we tend not to leave them that long. There'll be little green sort of spheres. And well, a fruit is a reproductive sort of structure from a flower. So you could argue that potatoes are fruit? No. No, because it doesn't come from a flower. It's asexual. All right, you don't need 2 potato plants in order to get potatoes. So no, and it doesn't count as one of your 5 a day either. Right? So no, unfortunately, potatoes are not. Curiously, though, sweet potatoes are, aren't they? But potatoes are not. So if you've got to eat chips, go for sweet potato chips. Anyway, I always said we weren't going to sidetrack. Right. Okay. So now the other thing that's on here, you noticed as I tried to scroll up, of course, it decided that the ink was going to stay still and the page was going to move. So hang on, let me park that at the bottom there. If I now clear the ink, I can come down to the other bits. Hypothesis. This is a bit of a harder thing to do because it could end up being quite lengthy, but this is what we're testing. And it needs to be in the assignment somewhere in the introduction. And it's to do with the influence of the independent variable, that's the water potential, on the change in mass of potatoes. So in its simplest sense, I'm not going to try and get you to create one because it gets ever so tangled up. It's something like, but you need to sort of write this in a better form probably, placing potato pieces in sucrose solutions of different water potentials will cause different changes in mass. And then we need to say that there will be which is equal to the water potential of the potato, which causes no change in mass. Something like that. There will a water potential. There will a water potential be. There will. I'm going to put it in the wrong place. Be a water potential. All right, that's equal to the water potential of potato, which causes no change in mass. You could add, which can be read from a graph of the results. But you don't want your hypothesis to be a paragraph. The ideal hypothesis is only about a sentence. Water potential equal to WP, water potential. I could have put the psi. Of course, that would have been better, wouldn't it? This can be read off a graph of results. So something along those lines. Okay. Now, the next thing then is to look at the actual practical procedure. And although I've said it will be agreed, I'm going to tell you what to do. Like that. There is a sheet that will be available when you come in, and I will put this onto Moodle so that you know what's what. You haven't got it at the moment, but it looks like this. All right, so this will be the actual instructions. So these will be here in the lab when you come in. You will work as groups, and the 3 members of each group, team member 1, 2, and 3, will have different jobs to do at the beginning, and then you'll all have to muck in. But to get it set up is quite time consuming, so you need to work quickly, but carefully. So you need to be able to trust the other members of your group, right? And that is part of the skill. Remember when you're writing your UCAS statements, you keep writing things like, I work well as part of a team, and this is one of the ways we prove it. If fights break out, then heaven help you. Right, team member 1 is going to make up the sugar solutions, and most of you will look at that and think, I don't want to be team member 1, it's got numbers in it. It's actually very straightforward because I've told you exactly what to do. So you are given some 1.0 mole per decimeter cubed sucrose solution. It will just say 1M on it. You're given some water. You're given some measuring cylinders. Simple as that. We don't do this as accurately as we could do, but water potential of 0 obviously is just water, and we make up 50 centimeter cubed of each of these. To get the minus 260 kilopascals, we need a concentration of 0.1, so it's 5 of sucrose solution, 45 of water. To get the next one, it's 10 of sucrose solution, 40 of water. That's not difficult, is it? You've just got to make sure you label your beakers properly. Once you've got 50 centimetre cubed, you divide that between 5 test tubes, standing in a rack. Very important, test tube racks. Oh, I do apologise. Honestly. Yeah. And it said suspected spam as well. I love the way it does that now. And apologies to people listening to the recording. You've got your 50 centimetre cubed. You divide it between these tubes, but you need to keep track of them. So you label a test tube rack, your 5 tubes in there, and you just divide this solution between them. There's plenty to cover the potato, so it doesn't have to be accurate, as long as you just split it between the tubes. So you've got 5 tubes with water in. You'll have 5 tubes with each of these solutions in. So you end up with 35 tubes, all right? 7 test tube racks, 1 for each of the concentrations, each rack with 5 tubes. Hopefully we'll lay out enough racks and things before you come in, but you know, they're not hard to find. So team member 1, and you have to decide between you, team member 1 will be doing that. Sugar solution, water, divide it between 5 tubes, label your test tube rack, and that's that job done. Team member 2 is the potato cutter. Now, this person's got to be very careful to try and only use 1 potato so that you're reducing the variability. And you use, and I couldn't find them yesterday, so I have to describe it, a thing called a cork borer. A cork borer is a little metal tube with a handle that you can push into a cork to make a hole for glass tubes to go through. But of course you can also push it into potato. So the cork borer can be used to create these round sticks of potato. So you have to go as vertically as you can. If they cross over, you end up with chunks out of them, and that's no good. You want to get as many nice, clean sticks of potato as you can. At this point, we usually find that at least one of the potatoes is soggy in the middle. People pull the core out and it goes, yeah. So we will have a couple of spare potatoes about. You end up with these sticks of potato, and from that you've got to produce 35 close to identical bits. All right, so you get as many sticks as you can, and then you chop them. You always remove the skin. That would wreck things, wouldn't it? But you're trying to cut at least 35 pieces. We go for 10 millimeters in length, and that gets them as consistent as we can seem to manage. Now, they're not going to be identical, but it doesn't matter too much because we're going to work out the percentage change, not the absolute change. So 10 millimeters in length, and you end up with 35 of these. That gives you, all these bits of potato that can then be rolled on paper towel to just lightly block the surface moisture away. So that's what person 2 is doing, cutting the cores and then chopping them into 1 centimetre pieces, all right, 10 millimeters long. Do a few spares because you might find you've got some anomalous masses and you might need to replace some. Team member 3 is doing the weighing. Now, we have 3, and this is the reason we don't use more than 3 groups of you in here at once. We have 3 electronic balancers that will measure to 2 decimal places. 2 of them are those little ones, the little pocket ones, and then we've got 1 standard one. So those will measure to 2 decimal places, and you then have to keep track of the mass of every bit of potato, and you've now got to follow it. It says here, place it in the space on a laminated card. This, for those who can't see it, is a coloured piece of paper that's been laminated and divided up into squares, 35 squares, 1, 2, 3, 4, 5 across the top, representing the 5 pieces in each solution, and then the concentrations of the solutions down the side. So you place your piece of potato in an appropriate square, and you can write its mass on here. All right, so you can write the mass of the potato on the card next to it. But it's now been allocated a place and a tube, if you like, because your test tubes will all be organised. in order. And you now know exactly, and if I say, right, where is potato piece number 3 in minus 860 kilopascals, it's that one, and we know which tube that's going in. So you've now got a way of following your 35 bits of potato. All right, this was a stroke of genius on my part, I have to say. As you go through this and you're weighing, doing the initial weights of your potato, you might well find you've got some anomalies that just don't fit in. They might be much bigger or much smaller than the others. You know, if you're measuring, you've got 0.35, 0.37, 0.39, 0.5. That's probably a bit over the top, that one. You either trim a bit off it or you take another bit of the potato. Try and get yours as consistent as you can. So you've got 35 bits of potato sitting on this laminated card, all weighed. If you get to that point before the solutions have been finished, you muck in and help with the solutions. So that's our 3 team members, all right, sorting things out. So team member 1 making solutions, team member 2 cutting potato, team member 3 weighing potato. But obviously you make sure that you get this done as quick as you can. At that point, we then have to get the potato into their tubes as quickly as possible, but you've got to get the right ones in the right tubes. So this, you have to be organised. Or somebody in the group has to be organised, all right? You might want to appoint a manager. But you've got to make sure that you are putting the right ones in the right tubes, otherwise the masses afterwards won't make sense, will they? So you put them in their appropriate tubes as quickly as possible. Give them a little shake, because some will float and some will sink. That's related to the strength of the sugar solution. And you leave them for an hour. Careful that you time it, you know, as close as you can. I mean, a minute here or there isn't going to matter. What do you do in that hour? Well, they could do with a couple of shakes perhaps during that time, just to move them about in the solution, but bring something to do. A book to read, a puzzle to do. Last year, one group brought in Cards Against Humanity. So, yeah, that was an eye-opener. I learned a lot about that group. You can, you know, you can bring what you like to do in that. You know, it might be laptops and get on with some work, and you can be getting on with anything else. Yeah, you can create yourself, between you, a spreadsheet for doing the end calculations if you want. You know, that will do the change in mass and the percentage change. That you can sit around somebody's laptop and do that if you're organised. So, yeah, you can work on other parts of the assignment. You can sort out a results table. And then at exactly an hour, obviously you can take a break as well. Go for a walk if you want to. You take each piece of potato out of its tube. How are you going to do that? How are you going to get them out? Use your fingers. Literally put your fingers over the end of the tube, tip the solution down the sink and drop it on a paper towel, because you don't want to handle it, do you? Because that would squeeze the surface. So you tip the solution down the sink, you've got your piece of potato, shake it onto a potato towel? Weirdly, I said that yesterday as well. Paper towel. Dry it the same way as you did before, back and forth. Weigh it. Record its new mass, calculate the absolute change in mass, so that's the number of grams, you know, it might be 0.5, 0.26 or whatever. Make sure you record, well, to start with, yeah, record its new mass, and then you record the change in mass, making sure you get positives and negatives right. Really, really important that you get plus and minus right. You're then going to calculate your percentage change in mass, and it's got to be plus or minus, all right, which is why we've got these little symbols, plus and minus percentages. And I will set up a spreadsheet like the one I showed you yesterday for you to put your results in before you go. Job done. Right, that's the practical. So you can see why we have to be really prepared for this. But the amount of data we manage to gather through this is so good. You know, getting 55 pieces of potato in each sugar solution is pretty good. We used to do it with just one for each group. You know, you'd put a stick of potato in, and that's just not enough. You know, you'd have 11 sticks of potato. No, you know, if you're looking at real scientific research, you are not going to be looking at just a single amount. So, all right, at that point, we have created a spreadsheet. That, when we've got everybody's results in, which will be at the end of your Thursday morning session, won't it? I will then sort of tidy it up if necessary, and I'll stick it on Moodle. And at that point, you will then be able to do the mean. Oh, you need to find the anomalies and then do the mean values without the anomalies, standard deviations without the anomalies, a graph of the water potentials against the mean values, put the standard deviation error bars on there, and yeah, there you go, all sorts of things to do. And you might be able to start that next Friday afternoon while I'm around, although I can't give a lot away because it's a graded thing. Now, let's get rid of that and go back to what, the instruction sheet. The instruction sheet's not yet on Moodle, but it will be. And I realise I've just closed the assignment down, and I need that because I want to go through what's to be handed in. I will put the instruction sheet on Moodle so that you've got it. It's good to come in with a mental picture of what you're going to be doing. Whichever member of a team you are, it doesn't hurt to know what everybody's going to be doing, so that you are much more efficient if you are prepared beforehand, aren't you?
Will you tell us our teams beforehand so we can then?
No. No, no. I'll sort the teams out literally just before you come, and I'll write them on the benches. What I usually do is I get a marker pen, not one of those because that's a permanent one. Yeah, let's take a blue one out. I just write the names on the benches. So yeah, because it might depend on, if anybody is off sick, then we have got an issue, obviously. But no, I mean if it's well, the way things have been going recently. But hopefully everybody will be in. If not, if you are ill that day, you're going to have a problem because you need 3 people to do this, and you are graded. Well, in fact, it's one of the assessment criteria is that you do the practical, so you'd have to do it. How would you do it? Well, it is possible, but you would have to do the whole lot yourself. But we would do it just with single pieces of potato, rather than doing the 5 bits of potato in each solution. If you miss this, you will do the whole thing yourself, but with a single piece of potato in each solution. I don't know if I showed you last year's results that had the name Hannah at the top. Hannah was the one who couldn't be in the classroom, so she couldn't work in a group to do the practical work. And she came in and did it herself. She actually did 2 separate pieces of potato. She had enough time to do that. But she managed to do it on her own. So it is possible, but the best way to do it is obviously to be here. And of course, if you aren't here, then you leave a group with 2 people. So, yeah, that can be an issue. If we do have a group with 2 people, then either Jill or I will make up the solutions. Okay. We've got to do that for one of the science groups anyway, because there's 14 of them. Yeah, so they've got 3 3s on the first session, and then a 3 and a 2 on the second. So the 2 will get ready-made solutions. Okay, now let's have a really good look at this, because if you don't follow this to do your write-up, you'll be lucky to pass. And as far as grading's concerned, it's how well you do this is going to make the difference. So I've given a huge amount of guidance here, probably more than I'm supposed to be absolutely honest. What I do that I think tutors in other colleges don't do is to put it in the assignment. I'm pretty sure that tutors in other colleges will go through this sort of thing. But I'm very open about the guidance that I give, hence it's in the assignment. There may be more guidance required. I have got other things on standby, a bit like the first assignment. We'll see how it goes. Now, you have a title. This is Cells Assignment 2, should be the first thing you write at the top. You set your line spacing to 1.5, which needs then to be checked afterwards. It's amazing how many of you have handed in pieces of work where the line spacing's gone partway through. And you put your name in a header at the top right. Okay, it's the same rules each time. So, you know, that from the first one, your name, your page numbers, your line spacing, all that sort of thing, it always applies. All right, it's a standard presentation. You don't lose grades on that sort of thing, but it's getting you into good habits, because when you go to university, there will be standard styles. It won't necessarily be mine, but certain tutors will say, I want you to do it like this, so I just need you to get used to doing as you're told. I hope it's at least a sensible way. Now, you have a title, which you could just put osmosis, that's fine. And then you write the word introduction. This is a practical report, so it's structured like that hand-drying research paper. You have sections with subheadings. And the first subheading is introduction. We're not doing an abstract for this, but we are doing an introduction. And your introduction, like with any other introduction, sets the scene for osmosis. In order to do that, you've got to talk about cell membranes. A diagram can do a lot of the work for you. So you need to describe the structure of the cell membrane. You also need to describe different types of transport across the membrane and why they're necessary. So, for example, small molecules such as oxygen can pass through the membrane by simple diffusion. Charged particles such as sodium ions can't pass through the non-polar part of the membrane, so they have to have channels. They have ion channels, which are proteins, and that's facilitated diffusion. So, you know, you're explaining like this about the different types of transport related to the structure of the membrane and the different sorts of particles. Water, which of course is critical to all cells, can only pass through the membrane by, you know, wiggling through in very small amounts. Most of the water that passes through the membrane goes through protein channels called aquaporins. This is all in your notes. And the process is called osmosis. You then define osmosis, and you can use a diagram like the one that's in this assignment. And as you're writing this, fully referenced, so you put in citations. A citation in your work is just the author name and the year of publication, nothing else. Now, you might use a particular author's name. I can't imagine you would, but at the end of sentences or paragraphs in brackets you can put author, comma, year. So that's the main bit that's going to get you through assessment criterion 2.1, is relating the structure of cell membranes to the movement of substances across them, but emphasising osmosis because that's the one that we're looking at. The next bit is less critical in terms of the assessment criteria, but you outline the aim, which is really just a sentence. You know, we're going to stick, no, you don't put we. Some potato will be put in different solutions. We'll see what happens to the weight and, or the mass. Sorry. No, definitely not weight. No, you don't personalise ever, do you? Yes. Oh, did I? Ah, okay. Definitely not using the word we. It's still in the introduction, really, yes, that you say what the aim is, to find the water potential by using bits of potato in different water potential sugar solutions. Say what the variables are, literally copy it out of this sheet that you've just written on. State a hypothesis. You might predict if you want to. Keep that brief because that's not really getting you anything. And similarly, the next section to have a subheading, materials and methods, that is really brief. The list of equipment used is things like potato, sugar solution, test tubes, knife, scales.
We can bullet point it, can we?
Yeah. The more important to bullet point is the actual sequence of activities. Cut up some potato, make some solutions, leave the potato in the solutions for an hour, weigh them before and after. You know, don't write it out in a great long couple of paragraphs, bullet point or numbered point, like a recipe. I would prefer for this, for the method, I would say for this it should be numbered because it's sequential, isn't it? You don't have to, and this isn't assessed. This is just linking the introduction to the main bit. So this should be really quick. You then have the results subheading. You put a table in which is not every single 385 piece of data. All right, your table looks something like right, you'd have the mean, and you'd have the standard deviation, and you would have your water potentials across the top. All right, so we've got 0, minus 260, whoops, minus 540, was it? Something like that, and so on. So this is the data that's going to create your graph. So that huge table with the 385 bits of data in can go in your appendix if you do one. All right. It's worth having because you can actually identify your anomalies on that. But you don't want it in the results section. It's just too much. It's, you know, that much data is not meaningful. All right. You might want to put see appendix for full data set. Yeah. Yeah. No, after. Yeah, because the appendix isn't marked. It's just, you know, referred to for sort of completeness, if you like. So you also have your graph. You have to put your table before your graph, of course, because your graph is your, you know, what you get from your table. Get those the right way around. Make sure you've got sensible titles, grid lines, error bars, all that sort of fancy stuff on there. As beautiful as you can make it. So that is the results section. You might want to, in here, refer to your full data set. Oops, full data set in the appendix. E.g. for the anomalies. Because the mean and standard deviation are calculated after the removal of anomalies, aren't they? So, you know, to show the data set and the those spotty graphs, yeah, for picking out the odd results. So that's the results section. All right, it will have a subheading. Right. Now, so far, you'll only have covered 2.1 is done in the introduction. 2.2 comes in there. So we haven't covered much yet. The other 3 assessment criteria are all covered in the discussion. So your next subheading is discussion, and this is where you use the majority of your words. In order to get 2.4, you are going to refer to your graph. You're going to give an estimate of the water potential from that graph. Might use a snip if you want to, like we did yesterday. And you explain the theory about osmosis, you know, that when you put potato in a hypotonic solution, it takes up water and gets bigger. When you put it in a hypertonic solution, it loses water and gets smaller. but there's this point on the graph where it crosses the x-axis, which you are assuming is the water potential of potato tissue, and then you compare with Kaczynski. Now, referencing Kaczynski is tricky because he is quoting a lot of other people's data. The biggest problem you'll find with Kaczynski is that the water potentials are not expressed in kilopascals, they are expressed in bars, but they are very easy to convert. You just multiply bars by 100 to get kilopascals. So if he says it was 5.4 or minus 5.4 bars, that's just minus 540 kilopascals. All right, but he reports a lot of other people's work. Now, you can't read their work. We haven't got it. So if you want to refer to any particular people that he's written about, you have to use what we call secondary referencing. And I'll cover that after we've done the practical work. You know, if you've got, I don't know, Smith and Jones have reported that they found that the water potential of potato tissue was minus 540. You can say Smith and Jones, 1976, in brackets, cited in Kaczynski 2012, found that it was minus 540. All right, so that's secondary referencing, that you say the first person's name and the date of their publication, cited in Kaczynski, 2012. Alternatively, you can just sort of gather together and not refer to those individual people, take the easier route and say Kaczynski reports a range of different water potential values achieved by other investigators, including, and then just give some examples. All right, as long as you are saying where you've got that from, that will do. But if you want to do the secondary referencing, obviously, it's a good skill. Kaczynski's already on Moodle, so if you want to have a look at that beforehand, you can do. All right, but remember, you need to multiply by 100. Now, there's a lot of technical background stuff on the Kaczynski paper about different techniques and theories about osmosis. You don't need that. All you want is the results. So there's only a couple of sides that have got results on. But he also refers to things that might affect the outcome. Things like the weather when the potatoes were growing, different sorts of potato, white potatoes, red potatoes. The solution that's used, do you use sugar? Oh, sorry, do you use sucrose like you are, or glucose, or salt? Different results for that. So he's got some quite useful stuff in there that I've not managed to find elsewhere. If you do find it elsewhere, that's great. But this is just a particularly rich source of data to compare with. So to get 2.4, that's really quite a substantial piece. Okay, drawing your conclusions and comparing with values from Kaczynski. Absolutely critical that you do that. To get 2.3 is fairly straightforward. This is a paragraph. The significance of using mean values, remember that's where the word accuracy comes in, that you are taking the mean of 55 bits minus the anomalies. And so that improves the accuracy. And explain what the standard deviations show. They show the spread of the results and therefore the precision for each solution. And so that's sort of the basis of what you write there. And if you comment on things like, you know, how big the standard deviations are in different parts of the curve, that's improving it. I will be going over this again, don't worry, you haven't got to remember it all. It's really hard to actually try and understand what you've got to write up when you haven't done the practical. And then discuss the success of the investigation for the last bit. Consider aspects of the method. You know, if you think there were aspects of the method, that's the general method, not what you did. It's not personal failings in here. All right. If you think we should have measured the concentration of the solutions more accurately using pipettes rather than measuring cylinders or something like that. comment on anomalous results. Are there any sources of error and bias? You know, we couldn't use one potato for everybody. You know, there was variation. That sort of thing can go in there. Somewhere in all this, you've got to say whether your results support the hypothesis. I think that goes in there as well. But make sure you've checked the assessment criteria. And then your references at the end, because all this end section is going to be full of references. Anything that you've used anywhere, including for images, must be listed. Several people in the first assignment haven't put references, haven't listed the full source for the sources on their images. But everything that's used in your work has to be listed at the end, and everything listed at the end I must be able to find in your report. All right, so we will come back to referencing for that. Okay, so next week then is all practical, and then the week after that has got a bit more maths, but I will have this session of support where I particularly go over Excel and referencing in relation to that. All right.
Can I just ask,
You can
after
ask.
referencing, you said about an appendix. Do you
Oh,
need to do that?
you can. After references, you can put in an appendix. The appendix can contain anything that's not actually going to be marked. It might be that full data set. It might be the graph with the dots on it for identifying the anomalies. It might be the dilution table off the instructions for making up the solutions, because you don't want that in your work. There's anything that's not really going to be marked, but is useful to refer to. For that, you could say, make up solutions, see table 1 in appendix or something like that. So it's a useful addition. All right, so don't panic over this. I know it sounds huge. You can do it in bits, and there will be a lot of support. All right, so I will stop at that point.